All right, guys, it's Thursday afternoon, snow day, spring semester, man. Okay, hope y'all having a nice day, chilling. Uh, I thought I'd come in this afternoon, actually. I chilled out this morning and walk through the rest of chapter 17. Guys, I'm going to send out an email in a minute. You guys got to watch these lectures. You're not going to get enough just sitting there meeting once a week in class. So I'm going to finish this up. I'm going to finish this show. This one will finish chapter 17. And get us caught up, and we'll go to 18 on next Monday. All right, I'm going to share my screen. And I think this is about where we stopped at on Wednesday. And I believe we were looking at the heart parts, noticing that they always cut this kind of weird. Notice the left ventricle is always thicker than the right ventricle. But this left ventricle is, if you measure it, much bigger than that right ventricle. But for some reason, it doesn't always work out that way. Now, next week in lab, we're going to actually go to blood. Those that are missing lab today, we will just go into blood next week. And then 2 weeks from next week, 2 weeks from this week, we will do the heart. We'll actually cut the heart. Okay, but you can see this is the cut we're going to try to make, where you can see, remember, 4 is a while, 4 chambers, right atrium, left ventricle, right ventricle, 4 blood vessels, vena cava coming in, pulmonary trunk going out, pulmonary vein coming back, and then the aorta taking the blood out. Okay, so we have, and then the 4 valves, right? Bicuspid over here, tricuspid, pulmonary seminal valve, and then aortic seminal valve up under here. Be sure and watch those videos. They're excellent in the study area in your lab, or in the lab book. I think we shot one there. We went through the blood. Okay. Now I get the slide to move now. Here we go. All right, here I think we looked at this. I'm still reviewing a little bit from last lecture. Here's the cadaver. Pretty crazy, real heart picture there. Okay, and we've already talked about the valves, right? And we talked about the chordae tendineae, which is the straps that prevent the valves from black flow. And you can see it right here, and you can see it in the picture right here. So when the blood comes down here, and then the ventricles contract, it's going to want to go back into the atrium. And these flaps prevent the flaps from only from going back the other way, letting blood go back into the atriums, same as over here. So these chordae tendineae anchor this to papillary muscle in the bottom of the chamber of the ventricles, and it's kind of like guide wires on a light pole to keep it from falling a certain way. And this keeps the valve from blowing back open into the atriums, allowing blood flow to go back into the atriums. And the papillary muscle is the muscle that the chordae tendineae is attached to. Okay, and we talked about that. Okay, we went on. I'm going to move here inside the look from the top. If you make a cut here and look, here is the pulmonary valve closed on the left, and here it is open on the right. And then we kind of walk through the blood flow again. I'm not going to bore you with this, but you can walk through. Remember, we did it in class and we talked through it, but remember, blood starts in the what? Go back to this picture and walk through it if we want to. Blood comes from the body into the vena cavas, into the right atrium, through the tricuspid valve, into the right ventricle. Then when this side contracts, the blood goes through the pulmonary seminal valve to the pulmonary trunk. Then it goes to the lungs, does the gas exchange, comes back in the pulmonary vein into the left atrium through the bicuspid mitral valve to the left ventricle. When this contracts, it goes out the aortic seminal valve to the red aorta tube, which is over, the aorta is here, and the other one's on top of it. So pulmonary trunk is on top of that aorta. You can see that was a pretty bad example of my arm, sorry. But anyway, then in lab, we're going to learn 4 of the structures in the ventricular septum, okay, and chordae tendineae, papillary muscle, and then we'll just lump all the coronary blood vessels together, and we'll talk at them, and we're going to look at them in just a minute. The 4 big ones that supply blood to the heart, okay? Okay, moving on, and so we got that. That's the blood flow as I give in class, but there it is written out for you there, and it's on the other lecture. Couple of heart valve problems. Look at this, insufficient, the valve fails to close properly and you get leakage back in that we were just talking about. And stenotic is if it gets calcium deposits and it gets hardened and it won't open, it's not flexible enough, so it's not letting the blood go through properly. It's pretty famous. And of course, heart murmur is anytime you hear an extra sound in a heartbeat with a stethoscope, and that could be to a valve not closing properly or allowing blood to go back through or just closing at the wrong time. So you should get that ba-boom, ba-boom, lub-dub, ching-ching. And I always laugh, never heard lub-dub myself, but ba-boom. Okay, and we'll talk about that. The first sound is the what? The 2 Atrioventricular valves, bicuspid and tricuspid closing, and the second sound are the two similar valves closing. Okay, so the next thing we're looking at is how does the heart get its food? And I went to talk about this in the class the other day. The coronary arteries come off the aorta, off the ascending order, and there's a right-left coronary artery, and they're going to go down into the heart. Okay, then there is the marginal artery. Let's just take a look. Let's get the picture and look at this. So here it is. You can see this is looking at the front of the heart. The left coronary artery comes out and comes down and branches down and covers the left ventricle. And then the right coronary artery comes down and branches into a marginal artery, and it goes into the right ventricle. And notice that the left coronary artery branches down, and once it branches, it becomes the anterior ventricular septum, and that was real important, right? Very cool. So that's how they get that's how they get the blood on this side. And on the back side, you have there's a branch coming off the right coronary artery called the posterior interventricular artery that comes down. And you can see there's another one comes off here called the circumflex. Okay. And so these four major blood vessels then go down and form into the capillary beds in the myocardium. And this is how the heart cells get their nutrients. And then they drain, and the color there shows you which one it does it. The veins come back out, and there are three big ones here: great cardiac vein, small cardiac vein, and middle cardiac vein, and they all dump into this coronary sinus. And you can see it here looking. And like I said, this is one of the more weaker pictures in the book. I'm not real crazy about these. But here on the right side coming off the right ventricle is the small cardiac vein here. And over the left, which is bigger, is the great cardiac vein with this anterior ventricular vein coming up into it. And on the back, we have that coronary sinus posterior vein of the left ventricle, and then the right there, and then we have the middle cardiac vein here. Okay, so those are just, we probably won't label those on test, but just kind of know those, but not a big test question, but just, I'll let you know what pictures we have, okay? Plaque buildup, this is a big one, big cause of this, and this has been a kind of an interesting thing. I think one of the misleading things about this, we'll see in just a second, is this is a field of coronary artery disease. Is one of the main causes, is the leading cause of death worldwide. A cardio artery disease decreases blood flow to the myocardium, resulting in inadequate oxidation. And then when this happens, the heart will stop beating, and you will experience chest pain and stuff as this starts to happen. Okay, but the thing is that I think is very disturbing here is it's not actual plaque building up in the lumen. It is actually plaque building up in the artery itself that then pushes back out and closes it off. Look at this. Most dangerous potential is myocardial infarction, which is what we call a heart attack. I believe we have a picture of that. Risk factors of this include smoking, high blood pressure, diabetes, high levels of lipids in the blood, obesity, being over the age of 40 for males and 50 for females, and then there is some gender and genetics play a big role. Here is an angiogram where they shot a dye in here, and you can see the left coronary artery circumflex and anterior, and if you see a blockage or anywhere in that white line it turns dark, then that would be a blockage, and they would want to put a stent in there. Used to go in with angioplasty and rotor this stuff out, and they don't really do that anymore. Now they mainly do the stents, put stents in, or do a graft an artery from another part of the body and bypass the clot. Okay. Anyway, I thought we had a picture of an artery, but we do. I think it's in the lab. In the lab, we have a picture of the artery, and you can see it's actually inside the laters where the fat builds up, and that pushes and closes it down. And when it ruptures, then that would be a floating aneurysm, which would be really bad. Okay, now we're going into the second half of this chapter in 17.3, cardiac EKG, electrophysiology of the heart, and this can be a little bit tricky, so we'll try. I'll do my best, and we'll go through this. So, heart does not require conscious intervention to cause a heart to contract. Cardiac muscles are autorhythmic, have autorhythmicity. They will beat on their own. And one of my favorite pictures one time I saw in a video where they had like a heart muscle beating like this, and another one beating a little slower, this one fast. They pushed them together, they connected their intercalated discs, and they were beating in rhythm in just a few minutes. Cardiac muscle cells contract in response to electrical excitation in form of action potential, like skeletal muscle, but they also have the ability to generate their own action potential, and skeletal muscle don't do that, right? Cardiac electrical activity is coordinated by a very small, unique group of cardiac cells, muscle cells called pacemaker cells. These cells rhythmically and spontaneously generate an action potential that spreads to other top parts of the heart muscle to the contractual cells, okay? Of course, these are striated, and we looked at them in lab, so we know they're cross striations, and they have high, like lots of oxygen carrying, oxygen storing myoglobin, right? And they have those intercalated discs that we saw in lab, and those are very important cell junctions that allow the cells to communicate with each other with ions, and that's how we get that rapid cardiac contraction quickly. Desmosomes hold the cardiac muscles together, and gap junctions allow those ions to communicate, so we get that full heart rhythmic contraction that we saw in the video. Here it is. You can see the intercalated disc, and here, right there, the little white lines here, and you can see that allow things to go in from cell to cell. Can blow that up a little bit. Probably would have been nice. Now, like skeletal muscle fibers and other cellular cells, cardiac muscle cells contain selective gated ion channels in the sarcolemma, which is their cell membrane, right? And opening and closing of these ions is responsible for both the pacemaker and the contractile cardiac action potential. All right, now I'm going to try my best here because this is kind of wild. So, the different types of gated ion channels in the cell membrane. Voltage-gated sodium channels open in response to electrical charge across the membrane and all cardiac muscle except the pacemaker cell. Calcium ion channels demonstrate voltage gate opening, but timed gate closing. So they close after a certain period regardless of voltage. And all types of cardiac muscle cells have one or more types of potassium ion channels. Some are ligated, means something binds to them, then they open. Others are voltage. When the electricity hits them, they open. Okay, remember we already talked about voltage, the electrical difference between 2 points, and there is a resting membrane potential of minus 60 to 90 millivolts across the cell membrane. Remember we had that action potential in the final on the AMP1. Remember, the flow of ions or electrons with chemical cause creates a chemical gradient, and depolarization occurs when the membrane becomes less negative, right, and goes positive. And when that happens, we get we get a chain reaction that opens up all those channels all the way down, and so we get a movement of electrical charge along the cell membrane. And then it returns to normal, to its negative resting membrane potential, and that's called repolarizing. So when they get excited, they depolarize, and they get an action potential, and then they repolarize back to normal. Now, we're going to take a look at this. Remember that we like sodium on the outside, potassium on the inside, and we have that sodium-potassium pump that maintains those levels. Now, how do these pacemaker cells work? Pacemaker cells undergo rhythmic, spontaneous depolarization that leads to an action potential, and we're going to walk through that on these next couple of pictures, okay? Action potentials are transmitted from pacemaker cell to contractile cells through intercalated discs that unite them. Gap junctions in these discs allow electrical activity generated by pacemaker cells to spread to all cardiac muscle cells through these electrical synapses. And this is where we get this electrical synapses we talked about. More common is what? Neurotransmitters in that chemical synapses. This permits the heart to contract as a unit and produce a coordinated heartbeat. Reason cells of the heart are sometimes called functional synostomes because they are multinucleated cells, whatever. Okay, now let's look at these. Now, pacemaker cells only make 1% of all the cardiac muscle cells. 3 populations of these cells in the heart are capable of spontaneously generating an action potential, therefore setting the pace of the heart. 3 cell populations are collectively called the cardiac conduction system, and we're going to see them now. Now, pacemaker potentials. Much different than that of contractile cells. Depolarization of pacemaker cell occurs much more slowly, due in part to a lack of voltage-gated sodium channels in the pacemaker sarcolemma or cell membrane. The pacemaker cell action potential lack the plateau phase, and membrane potential oscillates, never remains at resting level, instead occurs in cycles, which last, with last event triggering the first. They occur because of non-specific cation channels unique to pacemaker cells. Now, I've read all that, but let's take a look at this in the picture in just a second. The slow initial depolarization phase that pacemaker cells start with the plasma membrane is hyperpolarized. That is the start of it. Opens these certain cation channels in the membrane and allows sodium ions to leak into the cell and potassium leak out, resulting in a slow depolarization to threshold. Then we reach full depolarization, then we get that. Then when that happens, the voltage-gated channels, ions open, and you get a much quicker fire, fully depolarized. Okay, now look at this. This repolarization, then I'm actually going to go to this. I think this is easier to see, and I apologize, but then that talks about it has a repolarization phase and then a minimum potential phase before it can fire again. So watch this. This is actually makes more sense. Should have skipped to this. So here is what you're used to seeing: sodium channels, right? For muscle, skeletal muscle, action potential. We also have calcium channels play a role here, and here are these HCNs, right? And remember what that stands for? I believe those were talked about right back here. HCN, that's the non-specific channels, I believe. Did I get that right? Yeah, I think that's what that was. Okay, now take a look. Slow initial depolarization, more cations leak in than leak out. Remember, because we've got these leaky channels here, okay? In the plasma, so one more time, more cations leak in than leak out through these HCN channels and in the plasma membrane, causing the membrane to slowly depolarize the threshold. So now this is preventing a rapid change. So here we go, we're going positive now. At threshold, voltage-gated calcium channels open and calcium rushes into the cell and we get a full depolarization. Okay, but look how it's going kind of slow here to get over there. Then bam, now watch. Calcium channels close and the voltage-gated potassium channels open, causing potassium to outflow, and that should bring it back down quickly. Okay. However, potassium channels remain open and the membrane hyperpolarizes. This opens the HCN channels and the cycle repeats itself. So in other words, and what you're getting is that this slow right here, one more time. These calcium channels close and voltage-gated potassium channels open, causing potassium to flow back out and it returns to negative. But notice that this has a little more gap in it than we would see in the cardiac muscle, okay? Now, where does these conductivity cells appear at? Well, there's two big areas, the SA node and the AV node. The sinoatrial node's in the upper right atrium area, and it will have a intrinsic rate of depolarization of about 60 times per minute, or about every second. And that's subject to sympathetic nerves that will speed it up. Parasympathetic nerves will slow it down. Then there is the atrioventricular node located in the about halfway down from between the atriums and ventricles, and it has pacemaker cells that can fire 40 action potentials per minute. And then there's these fibers called Purkinje fibers that carry the electricity up into the depths of the ventricle. And they, if you were to have a heart attack and knock out your SA nodes and AV nodes, then these can fire and give you a 20 times per minute atypical pacemaker cell because it will not function very well, okay? And let's look at where these are. So here's the picture. Here's the sinoatrial node, which is the main pacemaker of the cell. Here's the AV node. So what happens is we get the electrical impulse here, it goes to the atriums, they fire, and then the AV node delays the electricity just a few milliseconds, then takes it down the Purkinje fibers down past this AV bundle here that can fire on its own, and it goes down to the tip and goes up those papillary muscles and all the way up. So the contractions and the ventricle starts at the tip and squeezes all the blood out. Okay, pretty wild. Okay, all right. Now let's see if we can see what we got here. So how is it doing that? Okay, it's going to talk about these sinus rhythm cells. Each population of pacemaker cells can potentially pace the heart. One that depolarizes faster sets the rate. Okay, so they each can do that. Okay, we'll leave that. All right, moving on, and it's going to talk under normal conditions. SA nodes generate action potential that spreads rapidly via gap junctions to the surrounding atrium. Impulses are conducted by specialized arterial conducting fibers to the AV node. This requires 0.03 seconds. Once it reaches that, the conduction slows considerably as a result. Low number of gap junctions between the AV nodule cells, and this is going to slow the fire down. This slows the conduction generally down, and the AV node delay is about 0.13 seconds. This allows the atriums to depolarize and contract, and thereby fill the ventricles before they contract. Also helps to prevent current from flowing backwards from the AV node into the AV and atrium. Okay. Then the action potential goes down the AV bundle to the right and left branch down the Purkinje fibers that we just showed you in the picture. Okay. All right, let's take a look. This might be actually a good video. Let's take a look at this
one. As you observed in the experiment, contractile cells do the work of the heart by contracting, but they need pacemaker cells to tell them when to do it. The pacemaker cells function like an officer giving orders to soldiers, telling them to move. Let's now look at how those orders are communicated. During a normal heartbeat, action potentials originate in the pacemaker cells of the SA node. The impulse spreads across the atria, both via the internodal pathway and through gap junctions between contractile cells. At the AV node, where cells are small and have few gap junctions, conduction of the impulse is slowed as atrial contraction proceeds. The electrical impulse leaves the AV node via the AV bundle and rapidly propagates through the bundle branches. Rapid conduction continues throughout the Purkinje fibers, depolarizing the ventricles. Ventricular contraction follows depolarization. The contraction ends as the ventricles repolarize and relax.
Pretty good. Okay. Now, I couldn't remember what this was because this was actually new when we did this book. So it drove me crazy. I had to look it up. So these HCNs are non-specific cation channels. And what this is showing you is how the pacemaker cell will fire. And that sets our rhythm. Then look, then over here, the cycle begins again because of these self-linking these HCN channels. Okay. Kind of wild. Okay. Now, we kind of went through that, and I think that's a pretty good overview of that. So what do pacemakers do? Pacemakers are instruments that have wires on them to take the place of errors in the SAAV nodes. Could be due to a birth defect, or it could be due to a heart attack. And so it's a rewiring of this electrical circuitry to make the heart contract. Okay, that's pretty cool. Okay, now we're going to show you how contractile cells work. I'm going to skip on over to this picture because this is one I was trying to refer to, and I was wrong a while ago. The last one looked like this, was just showing us how the pacemaker cells work. Now watch this. So cardiac muscle cells are going to fire a little bit different. Their action potentials are a little bit different than the skeletal muscle we were used to seeing. So take a look. Rapid depolarization phase. Voltage-gated sodium channels activate, and sodium enters rapidly depolarizing the membrane, thus creating an action potential. Go straight up. Sodium channels are inactivated now, and potassium channels open, and that should bring it out. Right? Remember, sodium in, potassium out, and it should go real fast. However, look what happens. Sodium channels are inactivated, potassium channels open, potassium leaks out, causing a small initial repolarization. Next slide. Then calcium channels open, and calcium enters as potassium leaves, and that prolongs the contraction substantially, like that, almost 150, maybe 250 milliseconds. Then eventually, sodium and potassium channels close, and potassium continues to exit, finally getting a repolarization. And so what you end up with is this cool thought here. Look, this sequence of events of contractile cells potentially resembles that of a skeletal muscle cell, with one important exception. If a cardiac action potential lasted only 1 to 5 milliseconds like skeletal muscle, resting heart rate would be 15 times faster than it should be. This plateau phase lengthens the cardiac action potential to 200 to 300 milliseconds instead of 1 to 5 in skeletal muscle, and that slows the heart rate, provides time required for the heart to fill with blood before the ventricles contract and pump the blood out. Here is that skeletal muscle quick refractory period, right? Bang, bang. And here is this 300 millisecond contractual period or depolarization that we see in the skeletal and the cardiac muscle. So to me, that's cool. And that's one of your short answer questions, right? And if it continues contraction, that's tetany, right? And we talked about that. So it goes on in the slide. Filament stuff again, I'm not going to go in that. And now our next one is EKG or EEG. Now, why is it called an EKG? Well, I believe the Germans invented this, and they don't have the letter K in their alphabet. The cardiac sound they don't have a C. So the cardiac sound is spelled with a K. And so we call it electrocardiogram or EKG. And the three waves, and there are multiple waves, and it's a lot more complicated than I'm going to make you know, but we're just going to go real simple here. So you need to be able to draw one and label the P, QRS, and T wave, okay? One of the most obvious chances in heart is when you have a disturbance in the EKG, and you can get these arrhythmias or errors. Take a look. We're going to see those in just a minute. So the P wave represents depolarization of the atriums. Okay, the QRS wave represents the depolarization of the ventricles and the repolarization of the atriums, which are masked by the big waves. And then the T wave is ventricular repolarization. So we're actually measuring the electricity that your heart's giving off, and it can be measured by putting the EKG on the wrist and ankles. So P wave represents atrial depolarization. The QRS wave represents ventricular depolarization, which is also masking an atrial repolarization, returning back to normal the atriums. And then the T wave is ventricular repolarization. Now there are all kind of gaps and spaces that can be measured on this, and but a doctor, a cardiologist, or doctor can look at this and tell if you have a atrial problem, if you have a ventricle problem, or you have a repolarization problem of the atriums or ventricles by having different abnormal EKGs. And here's some of the intervals. I'm not going to go crazy on them. RR interval, PR, QT. A lot of these can be measured. Goes on and on. There's another one. Okay, and here's a nice little picture showing you the P, QRS, and T wave. So from here to here represents 1 heartbeat, and it beats again. So this picture represents 3 heartbeats. Bradycardia is if the heart rate is under 60 beats a minute, which would be low. And tachycardia is if your heart rate is over 100. Okay, and that's really dangerous. Okay, right left bundle branch errors here, and fibrillation. Atrial fibrillation means that it's got crazy, completely out of rhythm, probably due to a heart attack. And this is where they would use a defibrillator to reset the EKG. To me, the defibrillator is the equivalent of popping a circuit breaker. If you ever blow a circuit in your house and you hit the circuit breaker button, that is the heart's way of doing that, by shocking it, and you reset the EKG and it starts back over. Hopefully, that's what you're seeing in the movies. Here's ventricular fibrillation, which is also immediately life-threatening. So was the atrial fibrillation. That can be quite dangerous, but this is one where you coding here, straight line, shock them. Ventricular fibrillations are bad, right? Moving on, mechanical physiology of the heart, the cardiac cycle. Okay, and that's 1 complete heartbeat, right? And we got a couple ways we can look at this. It's just showing how the blood flows through and how the valves close. And with a stethoscope, you can actually put the stethoscope in these different places on the body and listen to the valves, open and close. So here's kind of cool. You can look here to hear the aortic valve, aortic semilunar, here for tricuspid, here for bicuspid mitral valve, and here on the back here for the pulmonary valve. So, yeah, and then we've all been the doctor, and they take the stethoscope and they listen to your heart and your lungs. And a good doctor can learn a lot with just listening to the heart. But looking where they put the stethoscope can tell us what valve they're listening to. A murmur then would be any defective in a valve that makes an extra sound, an abnormal sound. S1, we already said that, and S was the first sound, lub dub, a lub of the lub dub, and then our S2 is the second sound. First sound, atrioventricular valves closing, bicuspid, tricuspid closing. Second sound is both similar valves closed at the same time. Third sound, S3, can occur just as blood begins to fill into the ventricle, right after S2. And then there's another sound, S4, that can be heard when most of the blood is finished draining from the atrium of the ventricle, just before S1. So there are 4 sounds you can hear there. Okay, and that brings us to one of the most famous words that you're going to be talking about in the next chapter and this chapter, and the term systolic and diastolic. Systolic refers to contraction of skeletal or cardiac muscle. Diastolic means relaxation. Okay. And we'll be looking at how all that goes into the neck fix. So when we do blood pressure in the next chapter, we put a pressure cup on the arm, cut the blood flow out the arm, and as we release the pressure on the pressure cup, the pressure falls until the heart overcomes that pressure. And that first sound you hear is systolic, the top number in blood pressure, and that would be the pressure exerted by the heart on the artery when the heart's contracting, because systolic means contracting. And then the last sound you hear as the blood rushes back in your arm is the diastolic number, and that is the pressure exerted on the heart, by the heart on the blood in the artery when the heart is at rest. So diastolic is the bottom number. Pretty cool. And we see high blood pressure when that diastolic number gets too high. Okay, and here's a cool picture. Look at this. This goes through the blood flow again, showing, I thought one of the cooler things here, look at that the atriums are responsible for the last 20% of the blood. 80% of the blood going to the atrium ventricle falls in due to gravity. So the atriums just cap off the ventricles. It's not a complete atriums fill up the ventricles, like a gallon container. They're much less than that. They just cap off the ventricles. Okay, and that's pretty cool. And it's just showing you how that goes, how that works. Nothing much. About 70 milliliters of blood pumped from each ventricle. 50 milliliters of blood remains in each ventricle. So it's always doing that. Pretty cool. Just showing how it feels. Nothing much here. And then here's some crazy pictures that kind of put it all together. I think they actually stick it all together over here on this picture. But look at this. It's pretty wild. If you look at this, I need to blow it up a little bit better. But here is ventricular fill. So here is the EKG, P, QRS, and T wave, 1 heartbeat, 1 cardiac cycle. Here's your S1, S2, the heart sounds. And this purple one is our aortic pressure in the aorta. It's going to go up, then it's going to come back down. Okay, then the green one is ventricular pressure, and then arterial pressure. And here's where the mitral valve closes, here's where the mitral valve opens, and then volume changes in the left ventricle, full, then empties, then refills. And that amount that's pumped out is the stroke volume. So a pretty cool little picture here showing all, putting it all together here, that you can look at on your own. Okay, let's see, pressure, flow. I don't know if I want to show this or not. Oh, we can try it. Let's see. Some of these are pretty good, and y'all may not.
Let's review what you've learned. The cardiac cycle consists of all the coordinated events that occur in the heart to pump blood during one complete heartbeat. The timing of systole and diastole depends on the electrical activity of the heart, which can be recorded by the ECG. The cardiac cycle is divided into 4 phases based on the position of the heart valves and ventricular activity. Ventricular filling occurs when the ventricles are in diastole. The atrioventricular valves are open and the semilunar valves are closed. Atrial systole completes ventricular filling near the end of this phase. Isovolumetric contraction begins as ventricular systole increases the pressure in the ventricles. The atrioventricular valves close, causing the first heart sound. Ventricular ejection occurs when the increase in ventricular pressure opens the semilunar valves and blood leaves the heart. Isovolumetric relaxation begins when the ventricles enter diastole. Ventricular pressure decreases, causing the semilunar valves to close. Closing of the semilunar valves causes the second heart sound. When the ventricular pressure becomes less than atrial pressure, the atrioventricular valves reopen. The cardiac cycle repeats itself as ventricular filling begins again.
Okay, like the little cowboy there. Okay, now the last little part of this, I'm not going to go crazy on the test on it because it gets pretty crazy, but do know that part about the heartbeat and all that. But look at this cardiac output. Our normal heart rate is 60 to 80 cycles, per minute. Okay, and the cardiac output, amount of blood pumped in the pulmonary systemic circuit in a minute. And so that one is, in one heartbeat, stroke volume thing here. And in the measurement here, it's around 70 milliliters is the normal amount. So to multiply that out, so 72 beats per minute, you basically pump 5 liters every minute, and that is the volume of the blood in your body. So you're pumping all your blood to the heart every minute. That's pretty wild when you think about it. Okay, and then stroke volume average 70 milliliters, may range 50 to 120 extra. Stroke volume may be difficult to measure directly. Often measurements called the ejection fraction tells us how much is coming out. Okay. Now another weird thing about cardiac muscle is a concept called preloading. Refers to the length or degree of stretch of the sarcomeres and the ventricular cells before they contract. And so what this means is that the more you stretch them, the more they're gonna pump blood, they're gonna push more blood out. Okay. And that's pretty wild. And this actually gets its name. It's called the Frank-Starling law. Okay, and it's a relationship between preload and stroke volume. According to this law, increased ventricular muscle cell stretch leads to more forceful contraction. So stretching causes more optional overlap of actin and myosin filaments in the muscle cell, enabling a stronger contraction and a higher stroke volume. This ensures that the blood discharge from the heart is equal to the volume that enters it. Particularly important during exercise because you don't want to pump too much out in one part or get too much from the heart and would cause major problems in the body. Okay, pretty cool there. So it just talks about its ability to pump, nothing much here. Afterload refers to forces that right and left ventricle must overcome in order to eject blood into their respective arteries. Okay, and so we can kind of look at all the factors that affect blood volume here. And I'm not going to get into all that. Nothing much. The very common problem to people that are overweight is enlarged heart or ventricular hypertrophy because of this having to pump so much. And the more overweight you are, the more blood vessels you have, you may develop an enlarged or hypertrophy of the ventricle. Okay, and that's pretty common in athletes too. Okay, Now look, I'm just going to cut to the picture of this. So how does the nerve do? Okay, remember the sympathetic nerves are going to speed up heart rate and also trigger the release of norepinephrine, which is adrenaline that's going to speed you up. Parasympathetic nerves are going to release acetylcholine, and they're going to slow the heart rate down. Okay, so that was one of the questions on the effects of each of the autonomic nervous system. The parasympathetic speeds it, slows it down. Sympathetic speeds it up. Okay, nothing much here. Other factors, just nothing. Let's get this. But look at all the things going into cardiac. Look at the top part. Cardiac output, preload, stroke volume, parasympathetic nerves, body temperature. Down at the bottom, hormones that cause fluid retention. It's going to raise blood pressure. Sympathetic nerves are going to raise blood pressure, and increased body temperature raises heart rate. So all of these are just the bottom ones lower, top ones raise it. Pretty crazy. Heart failure, we've already mentioned that. Anytime the heart cannot pump properly, right? Edema, swelling, pulmonary problems, you have, all that can happen. Treatments here, obviously, start exercising, quit eating garbage. There are some drug therapies that diuretics can make you help lose some fluids. And then ultimately, though, we're working on a transplant. So we have a lot of people needing a heart, okay, or a pacemaker to help the heart. So we still do a lot of heart transplant. First one was done in the late 60s, okay? Don't know if I'm going to watch that one. I may just skip that one. I don't know. That way you don't have to run it.
We have all felt our heart beating more quickly or more slowly, depending on our activity. Let's learn more about how heart function changes to meet metabolic needs, whether hanging with friends, walking to class, or running to catch a bus. Cardiac output is the volume of blood pumped out by each ventricle in 1 minute. Cardiac output is the product of heart rate times stroke volume, averaging 5 liters per minute at rest, whether lying down, sitting, or standing. Heart rate is the number of beats in 1 minute, averaging 75 per minute at rest. Stroke volume is the blood ejected by one ventricle with each beat, averaging 70 milliliters per beat at rest.
Okay, I thought I had a little bit. I thought I had a little bit more on that one. Okay, now, so anyway, it's kind of cool there to see that. All right, now, let me show you something cool. I'm going to stop sharing screen. And about 10, 15 years ago, this company came out with an artificial heart. So check this out. I had it open here somewhere. Where did it go? Oh my gosh. I'm telling you, I had it right here. Okay, I had it open here. Here we go. Yeah, there we go. I knew I had it here. So this is the AviCor artificial heart, if you can see it. So it is, notice it has 4 tubes to hook up for the what? Vena cava, right? Pulmonary artery, pulmonary trunk, and pulmonary vein, right? And aorta, right? So vena cava is aorta, pulmonary artery, pulmonary vein. And it is like a blender here. And the coolest thing about it is self-contained, smaller than our heart. It's put in your chest. It has a transponder like your Apple Watch is charged with. So you put a little transponder on it tonight to charge your battery, internal battery, and then emergency backup battery in the abdomen to get you another emergency couple hours. We put this in people now about 15 years, and it's continuous flow, so it doesn't stop. The heartbeat, the reason our heart stops and fires every 0.85 seconds is to give the heart muscle time to relax and rest a second, then fire again. So this thing continuously flows. So these people have to wear, some kind of marker on them, a necklace or bracelet that says, "Hey man, I don't have a heartbeat," because it continuously flows. And we run these in the lab at up to 10 to 20 years of running them super fast. So I think someday this will be what we'll see replacing the heart. First time you have a heart attack, we'll put one of these in and we'll cut yours off like cutting the TV off when you go to bed at night when you die if something else kills you. Isn't that cool? So this is an AviCor replacement heart. I thought y'all might enjoy seeing that, okay? All right, so I'm gonna try to stop this thing. Here we go. I don't know if y'all saw that. I hope y'all saw that. Okay, let's try one more time. Did y'all? Yeah, I guess y'all saw it or not. We shall find out later. All right, guys. Sorry, kind of crazy. It's been a day off. We're going to start in chapter 18 on Monday, and y'all have a good day.